out. Okay. So the adaptive body defense is basically, if we talk about the adaptive body defense, okay, we get the diagram. Let me tell you where we are right now. Okay. Okay, here it is. So we finished with the let me get the pen. We finished with the first line, we finished with the second line, and today we'll be talking about the humoral one, right? So this one, if you remember, I told you this is not included, and even we could not finish the humoral one, right? So you can say that like half of the humoral response, okay, will be there for the midterm, okay? So we are going to talk about adaptive body response today, and we're going to talk about the humoral immunity today, right? So what is the meaning of the word humoral, guys? I explained you this word earlier, when we were doing chapter number 9. What is the meaning of the word humoral? Body fluid, absolutely. Be it the blood or lymph or interstitial fluid, it's called as a humoral. Okay. So in the adaptive body response, if you remember, I told you the basic difference between the adaptive and the innate immunity is that in adaptive one, what do we have? The response from the immune cells is very, very specific, right? That means they know who is the intruder. That means they know who is the culprit. They know who is the microorganism, right? So it's very, very specific in the adaptive body response, right? So they know, okay, they have to produce a response against this antigen. Okay, this is the coronavirus which is in my body, right? So I need to produce response exactly for this coronavirus. That is why we call it a specific body response. And if you remember, I told you last time that the innate one is always ready to produce the response. It doesn't take a lot of time. It responds within few minutes, while as a specific one might take little longer. For example, if you have antibodies, and it might take 2 weeks for plasma cells to produce the antibodies, the activations and the proliferations, and then they start producing the antibodies. So kind of it takes a little longer time to respond, right? But it's very, very effective because it's antigen specific and it's systemic. What do you mean by systemic? That means it's not restricted to only one particular area which is being infected, right? The whole body will be, will try to show the response. The immune system will try to show the response to the entire body. Okay. And then we have the memory cell. That is something very, very important, which is not there in the innate immunity, right? In the innate immunity, there's nothing called the memory cell. Okay. But what is a memory cell? Memory cell will be, keeping the memories, storing a copy of the antigen. Okay. So that next time when we will have the same infection again, right? We don't have to start right from the scratch, if not right from the sensitization. We can immediately produce, for example, antibodies. Okay. The memory cells can divide to produce, plasma cells, and they can form the antibodies right away. Okay. So that is why having a memory of a particular infection is very, very important. For example, if the COVID will happen for the first time, right? So my immune system knows that this is the virus. Okay. So it has already stored the memory cells. So second time, after 2 months, 6 months, 1 year, if the patient is again re-exposed to the same virus, right, a patient needs not to be worried, because at that time the secondary immune response will be much more faster, longer, and very, very effective. Okay, we'll talk about that, about secondary immune response in a while in today's topic. So having memory cells are very, very important. So whether we are talking about T cells or B cells, both of them are part of the adaptive immune response, right? So both of them keep the memory cells. Okay, that is so special about the adaptive body defenses or the specific immune system. So, what are the types of the adaptive body defenses? One is the humoral immunity or the cellular immunity. So, immunity is of two types. Either we get the immunity, that means the antibodies are only present in the humor, they're only present in the blood, or present in the interstitial fluid, or present in the plasma. You will not have antibodies within the cells. Okay. So, that is the humoral immunity, which is provided by the antibodies. Okay, and then we have the cell mediated immunity, wherein we have the T cells, the T cytotoxic cells, the T helper cells, right? So all these cells will give us immunity. But here, if you remember, I told you last time also, the natural killer cells, okay, and from the non-specific one, and the T cells, right? The T cytotoxic cells, okay, they all are almost similar in basically, killing the target, right? If you remember last time, we also spoke about the natural killer cells can also, they not only kill the microorganisms, they kill the virus infected cells, right? So that if this cell is infected with the virus, so the whole cell will be killed by the natural killer cell as well as by the T cell. So T cell comes under the cell mediated immunity, but it's more specific than the natural killer cells. Okay, and your T cells can also kill the cancer cells. Okay, and T cells are the ones, whenever we take a foreign graft, for example, if kidney is being transplanted in a particular person, and if my body tries to reject it, not accept it, right, that is basically because of the T cells. Okay, we'll discuss all that in detail when we discuss T cells in detail. So our today's topic is mostly based on humoral immunity, the antibody based immunity. Okay, so before we start discussing anything, we first of all try to ask ourselves a question: what is an antigen? We've already always been talking about antigen, but what is this antigen? Okay, this antigen can be something, okay, which can induce a response in my body, an immune response in my body. It can be anything. It can be a simple dust particle, it can be a simple protein, it can be a simple nucleic acid, right? Animal dander, animal hair, any bacteria, any fungi, any toxic thing, any virus, right? So all these things can act as antigens in my body, which can stimulate my immune system to produce a particular response. Okay, so that's called an antigen. And then we have two types of antigens: the self antigens and the non self antigens. Okay, what are the self antigens? Self antigens are something which are basically present uh, on each and every cell of our body. Okay, let's say this is my RBC. My RBCs has for if my blood group is A, I know that on my RBCs I have the A antigen. Okay, similarly, all my body cells, whether it's an excretory cell from the digestive system or the epithelial cell, all cells of my body have something called as major histocompatibility, molecule. That is not in your book, so I'm just trying to tell you that we call it a MHC, MHC1 and MHC2, right? So we all have a kind of protein, you can say, on our cells, okay, which tells us that, okay, these cells belong to Miss Sabia. These are her cells. So these cells belong to me, right? So all like a tag, like the way you have a tag, the way you have a stamp, right? So all my body cells are stamped with something called as a self antigens. And when there is a training for the immune system, and we're going to talk about in a while about lymphocyte development, when we train the lymphocytes, to produce a response. So at that time, lymphocytes are taught that, okay, you're not supposed to produce response against your self antigens. They belong to you. So don't produce a response against those antigens. You need to produce a response only to the foreign antigens. So foreign antigens are, that can come from outside. Okay, and antigens usually have a large molecular weight. Okay, remember this molecular weight at 10,000, because after this we're going to talk about haptens, which are also antigens, but they have lesser molecular weight than the antigens. Is it clear? And whenever my body is not ready to accept the foreign organ, a foreign heart or a foreign kidney, right? It is basically because of these self antigens, because every cell of a body has the MHC molecule. It has a tag, right? It will not accept the foreign cell, because that cell is also tagged with certain amount of major histocompatibility, or self antigens we call them. Okay, then we'll talk about the haptens. If the question says that what is the difference between an antigen and the hapten, okay, what is the basic difference between the two? Remember that antigens have a molecular weight of about 10,000, while as haptens are something which are also antigens, but they don't behave as antigens, unless and until they combine with our body proteins. Yes, Shata, they're smaller in size. Very good. So haptens are something, as I said that, let's say this is a hapten. Its molecular weight is less than, as I said, that 10,000, right? And but this, its weight is lesser. You can say it's a little lighter than the antigen, but it cannot harm me at all. Okay, it can harm me only when it combines, if it enters in my body, like through my nose, through my mouth, and in my body, if it combines to the carrier protein. CP is a carrier protein, which can be any protein in my body, right? Then it becomes powerful, right? So so hapten plus carrier protein, then it becomes a complete antigen. Then it has, it has more weight, and it can behave as an antigen, and it can start producing a response in my body or activate my immune system, right? So my immune system does not recognize haptens, but yes, they recognize what? Carrier protein plus haptens together, which becomes a bigger molecule, and then my body starts producing the immune response. Okay, now what are the various things which can act as the, and remember, this carrier protein is our own protein, because when they enter our body, so our own proteins behave as the carrier proteins. Okay, so let's say what are the various things which can behave as an hapten, like for example, drugs. Some of the drugs, like your penicillin. Have you ever seen anybody, if the doctor has to give a penicillin to a patient, do you know that he basically tests the drug on his skin? We call it a scratch test. Do you know anything about it? Have you ever been tested for the penicillin? They inject the injection. They inject a small amount of penicillin under the skin, intradermal. Not actually. Yes, if a doctor has to give the antibiotic like penicillin, but he first of all tries to see, okay, if he is allergic to particular, if this penicillin can act as an hapten to this person. So before he actually injects the, if you're allergic or not, absolutely, we'll talk about hypersensitivities, hyperallergies, hypersensitivities at the end. Okay, but right now, so that means the penicillin can also act as a hapten for some people, right? It might bind to our proteins and then produce the immune system, start, activate the immune system. So that's why, there are many drugs, right, which are being tested before they are being really injected into the patient's body. So we check the particular person for any hypersensitivity. Hypersensitivity is, we're going to talk about at the end. Okay, so we do a scratch test on a particular patient, right? And if the patient is positive for the scratch test, then sorry, the doctor avoids giving penicillin injection. He might choose something else to which he is not allergic. Okay, then do you know something called the poison ivy? Have you ever heard about the poison ivy? What is it? It's a flowering plant. Most of the people are allergic to this poison ivy. It's a flowering plant. When you walk, if you brush with this plant, right, you might develop a skin rash, right? It's, because it contains a poisonous substance called as an orchiol, and that orchiol, when it goes into our body, it can behave as an hapten and produce the allergic reactions. Or sometimes the animal, some people are allergic to animal, right? Animal dander or animal hair, right? Or some people are allergic to detergents. Oh, I cannot use this body wash because, I get rashes after that. So that means that detergent is acting as a hapten for you. Or sometimes you're allergic to perfumes. Oh, I cannot use this perfume. The moment I use this perfume, I get a lot of sneezing. That means that perfume is acting as a, it's creating a hypersensitivity in your body, right? Or sometimes some people are allergic to gold. The moment they wear gold, they get a lot of, skin rash on their body. Okay, so that means gold for them act as a hapten. So all these are hypersensitivities. Okay, so we're going to talk about them at the end of the class, or end of today's class, end of the chapter, I mean. Okay, let's try to know what are the various cells of the adaptive defense mechanism. Okay, we already know that we have T cells and we have B cells. Now let's try to know how these T cells are produced, where do they originate from, right? If you remember, they also originate in the they originate in the primary lymphoid organs in the bone marrow, okay, from the hemocytoblast cells, okay, or lymphocytes. We spoke about them in chapter blood as well, the hemocytoblasts, right? So they originate from the red bone marrow, and let's see how do they originate, how do they mature, how do they actually become immunocompetent, right? Let's try to concentrate on this diagram, which explains it all. So I just said that whether it's a T cell or a B cell, both of them originate from the red bone marrow, from the hemocytoblast. Okay. So what is thymus? It is a primary or a secondary lymphoid organ, girls? It's a primary. No, 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 no. Thymus is not secondary. It's a primary. If you remember, I told you both bone marrow and the thymus are your primary lymphoid organs, right? So your yeah, so your T cells and B cells, they originate from the bone marrow, but then what happens? Your T cells, they move to thymus for to develop maturation, while as the B cells, they will stay in the bone marrow for their maturation. How you can remember is like B for bone marrow, B for B cells, T for thymus, T for T cells. That's how you can remember it. So once they're originated, what happens? They start, they go to thymus and they stay in bone marrow for maturation. T cells go to thymus and B cells stay in bone marrow for maturation. Okay. So what happens as they mature? What are the things which happen during maturation? What do you mean by maturation? Maturation means the T cells in the thymus and the B cells in the bone marrow, they develop immunocompetence. What is immunocompetence? Immunocompetence is, ability of our body to produce a normal immune response. That means your T cells and B cells are being taught how to produce the immune response in our body. Okay, so each lymphocyte, whether it's a T cell in the thymus or B cell in the bone marrow, they are exposed to one specific antigen over there. Okay, and then they become immunocompetent, right? And they kind of develop that unique receptor on their surface, and that is what they are specialized for. Okay, so it's something like if you have policemen, right? So before they actually fight with with the enemies, they are first being trained, okay, how to use the gun, how to climb on in the mountains, on mountains, how to climb on trees, and how to use this gun and that weapon, right? So they're being trained first. So immunocompetence means they're all being trained where? In the thymus, as well as in the B cells in the bone marrow and T cells in the thymus. Okay. Not only that, they're also taught how to differentiate between, okay, this is a self antigen. You're not supposed to produce response against this. And this is a non-self. So produce a response against this. Okay. So all that differentiation is also taught, that is part of its, training, part of its maturation. Okay. But remember, even they are mature, even they are being trained, even they are immunocompetent, but they are still naive. Naive means what? They're still young. They still don't know anything. Why do I say that? Because even though they are immunocompetent, they are mature, but they have not yet seen their real enemy. Once they will see their real enemy, once they will see the coronavirus really for the first time, then they will proliferate and differentiate. What does that mean? That means when they're matured, when they are matured in the thymus, from thymus they move to secondary lymphoid organs, both T cells as well as both B cells. They move to lymph nodes, they move to spleen, they move to mucous associated lymphoid tissue follicles, right? So as they move to these secondary lymphoid organs, but remember, even though they're immunocompetent, they're mature, but they have not encountered with an antigen. They have not. Why do we say naive? Because, for example, even though they are circulating in my lymph or in my lymph node, but they have not seen the real enemy first. That means the way, you know the policeman really fights in the battlefield. He sees his enemy for the first time when he's fighting. Same thing. So here they will be exposed, for example, to coronavirus. So as these cells, for example, this is an antibody, as it will be exposed to the COVID virus for the first time, that will cause its activation. Not only activation, that will cause its proliferation as well as a differentiation. They will multiply into effector cells and memory cells, which we'll talk about in a while, right? So that is, the final activation takes place when they finally encounter with the antigen. Did you all understand what is the difference between activation and the immunocompetence? Okay, great. Then we have, what are the other types of the cells? So we don't have only B cells and T cells in terms of adaptive system. We also have something called as an APCs. APCs are called as antigen presenting cells. They do not respond to the antigens, but they are equally important, right? Why they're equally important? Because they help in presenting the antigen to the T cells, for example. The three types of the antigen presenting cells in our body are your dendritic cells or the Langerhans cells. The macrophages also behave as APCs, antigen presenting cells, as well as the B lymphocytes. They can also behave as the antigen presenting cells. Now you might be thinking, what is an antigen presenting cell? What exactly, what is the meaning of that? So if you see here, do you remember, guys, the dendritic cell? We discussed that in chapter 4, skin. If you remember, it looked like something like this. Let me get a blue color. Do you remember in which part of the skin is present this cell? It kind of, looked like this with the long extensions. Does anyone remember which skin? Stratum spinosum. Absolutely. Very good. Absolutely, Rava. It's so this dendritic cells or Langerhans cell behave as the APC because it's the cell which will activate our immune cell, which will activate our T cell, okay, which will alert our T cell, right? Because you can think that, T cells, we have T cells, in our body, which we'll be discussing after the midterm. So T cells are basically those cells you can, in simple language, you can say that they cannot recognize the antigen. They're blind. In simple way, you can say that they're blind. They can't recognize the antigen. So they need somebody, okay, who can present the antigen to them, and your antigen presenting cells are doing that job, doing a presentation of antigen, to the T cells. So how do they do that? As I said that in your book, it's not mentioned about the MHC, the major histocompatibility molecules. Okay, so what happens? Usually these APCs have a molecule called as an MHC2. Okay, I'll tell you what happens. So let's say this is a dendritic cell, and let's say this is the antigen. The red one is an antigen. So it's engulfed. Antigen is inside the APC. I mean, inside the dendritic cell. The mechanism remains same for both of them. So we'll take an example of dendritic cell. Okay, so as it's engulfed, it becomes a phagosome. Okay, then this is kind of combined with something called as MHC. Just you can say a protein, something like that. Okay, so as it is combined, let me use another color for that. Let us say it is combined with a protein like this, which is an MHC, which is also produced within this cell. Okay, and what happens? So this MHC kind of, it displays the antigen on its surface. Look at this. This is all MHC. Usually on the antigen presenting cells, we have an MHC2. Okay, and this MHC2, what will happen? So the antigen is engulfed, but a small particle of the antigen is taken and it's combined here. Okay, so look at this. It's like, showing the red flag to the T cells. Oh, hello, I have an antigen. That means be active, start producing the response, right? So that is the function of the antigen presenting cell, right? Then your T cell will come, right? Your T cell will come, they will form a kind of a response like this. Okay, like this is your T cell. Like usually we have the T helper cell. We'll discuss all this, in T cells once again. Okay, that is how the T cell will get activated, because as I told you, T cell is kind of a blind. Okay, it needs the help of the antigen presenting cells. So the same mechanism for the, this was the Langerhans cells, and the same mechanism for the macrophages as well. Is it clear about APCs? Any question about APCs? So you have to know that how many types of APCs are there, and you have to know that what is their main function. Okay, as I said, that out of all these three, the most effective are your dendritic cells. Okay, very important. They can only hold pathogens and call, yeah, they can display, yeah, they can hold the pathogens, yeah. They can see, for example, APC like a macrophage. Okay, macrophage is also a APC, right? But what it does is it engulfs the whole antigen, right? It you know, it phagocytosizes it, engulfs it, but just take a small part of it and show it as a red flag. There's a danger here. Okay, so they can engulf them, they can act on the antigens, but then they take a small part of it and show it to the T cells that there is a danger, please be activated. Okay, so if you remember, I told you that dendritic cell is a part of an innate immune system, right? So this is like an innate immune system connecting the adaptive immune system, right? Helping the adaptive immune system. Okay, any questions about it? Are we okay till now? Okay. Now then we have the humoral immunity. Humoral, as I said, that humoral means, you know, body fluids. In body fluids, you will find everywhere antibody. Your antibody will not be found in the cells. Antibody you will find only, as I said, that you'll find them, for example, in different body fluids. We will talk about the different types of antibodies also maybe in the next class. The, you know, for example, in milk also we have antibodies, right? In blood also we have antibodies, right? The different fluids of our body will have antibodies. Okay, let's see how these antibodies are actually produced. Let's concentrate on the diagram. Okay, so look at this. We'll talk about the primary response. What is a primary response? That means a person got infection for the first time. That's a primary response. That means first time a person got, for example, nowadays we all are talking about COVID. So let's say a person got infected with COVID. Okay, for example, I got recently affected with COVID. That means that was a primary response for me. Okay, that means even though the cells of the B cells were produced, right, but they were not still activated, as I told you that they got activated only when they first time saw the COVID, right? That activates them. So you can say that it's the virus, it's the bacteria, it's the the foreign antigen which activates them for the first time. So if you see here, I have many types of the B cells. Look at this. It has a receptor here for green one, it has a dark blue one, it has a light blue one, showing that we have different types of the B cells, right? But for example, the COVID got attached to this type of receptor. So the moment COVID was attached here, right, what happened? Then this particular B cell got activated. As it got activated, it will start proliferating. That means it will start multiplying now. Look at this. I have only the receptor here is only blue one. Only this one got activated. So it will start producing more and more activated B cells. Okay. So as it will produce more and more B cells, then what will happen? These B cells will continuously produce the something called as what? The plasma cells. Okay, because these B cells, once they're activated, they become something called as a plasma cells, which are also called as an effector cell. So constantly all the B cells will become the plasma cells except one, except this. So this we are going to use as a memory cell to store as a copy, that when I will be once again exposed to COVID, I will be already having this memory cell. Okay. So what happened as the activated B cell, was converted into the plasma cell or the effector cell, and now it will try to gain more and more rough endoplasmic reticulum. Can anyone tell me why plasma cell has more rough endoplasmic reticulum? What do you think? Mm-hmm. Nobody? Chapter number 3 last semester. Having large amount of rough ER means what? What is the cell doing? The cell is active in what? Mitosis? No. Yes, synthesizing more and more proteins. Chapter number 3, basics. You forgot, guys. You can never forget the basics. Remember rough ER. What is the job of rough endoplasmic reticulum? To produce more and more proteins, right? So rough, having more ribosomes, having more rough ER, having more Golgi apparatus means that that cell is very. It's okay, Shata, no dear, no problem there. That means it's continuously secreting a protein. So why do they develop a lot of rough ER? Because now they have to secrete more and more antibody. Antibody is nothing like it's a protein. So plasma cells become large enough with lot of rough ER, lot of Golgi bodies, lot of ribosomes, which they will need to secrete lot of antibodies. Can you believe at an unbelievable rate of 2000 antibodies per second are produced in our body? If, for example, I got infected with the COVID, look at the rate of the antibody production. For 4 to 5 days, these plasma cells will continuously produce antibodies at the rate of 2000 antibodies per second. Do you understand? And then these plasma cells will die. And now these antibodies will be circulating in my blood, in my, for example, now since I got recently affected with COVID, that means right now I'm protected. Right now I have a lot of antibodies in my circulating in my blood, in my body fluids, in my saliva, in my interstitial fluid, in my lymph. So that is why I don't need a vaccine, because nowadays everybody is going for a vaccination, right? I don't need a vaccine because I don't need to worry at least for the next 90 days, 3 to 4 months, right? All these antibodies are going to be active, right? They're going to be continuously circulate. Okay. But what happens at the same time? One cell is kept as a memory cell. So what is a memory cell? Memory cell is that does not become a plasma cell. Okay. It stays for long. Okay. It is there for long-term immunity. Okay. Why long-term immunity? It stays there. For example, if next time, let's say after 1 month, 2 months, 6 months, if I'm again going to be exposed to the coronavirus, I will have memory cell, and memory cell will quickly get divided into the plasma cell and start forming the antibody. So the memory cells will divide directly into the antibody secreting plasma cells and producing more and more antibody. As you can see, this is secondary response. What is secondary response? That the person got infected with the same virus once again. And look at this. Just this memory cell will form so many plasma cells and so many antibodies. Look at this. In my first infection, okay, I just got these many antibodies. I had only these many antibodies. And the second in the subsequent infection, these many antibodies were produced. Look at the graph. Primary response has only these many antibodies, while as a secondary response have look at the amount of antibodies produced here. And not only more antibodies, even the time taken for the secondary response will be very fast, will be very less. Look at this. Here I started producing antibodies for, for example, after 1 week, antibodies produced, and they reached to peak after 2 weeks. Look at this. So all this time I was ill, I was totally sick. So that is why first time when you are infected with the virus, you become more sick, right? So, and it takes longer time for you to recover, because look at the time taken here. But God forbid, if a person is again reinfected with the same COVID, look at, there's no time taken. Within first day itself, you will start producing these many antibodies if it's a second time infection. So that is why secondary response is faster. okay, because the body does not have to go through the process of what? All this, the activation, right? The process of all these activations, sensitization activations does not happen here. Okay, so that is why the time period will be very less for the secondary response. Did you all understand what is the difference between the primary and the secondary response? And why is it so? Because just because of the memory cells, right? Because they will right away, they don't need to wait to, for all these activations and sensitizations. They will right away produce the plasma cells, which can produce the antibodies. Okay. So that is why for the second time, as I said, that the infection will be, it will not be, it will not be very harsh. Okay, it will be quick. The response will be rapid, right? And we'll also talk about IgG antibodies, which, that we'll talk about later on. What kind of antibodies will be produced? Okay, so that was about, okay, now this is the last topic which we're going to discuss, and we're going to stop over here. Okay, so active and passive immunity. So that means this is the last topic. We're going to stop over. This is the last slide which you need to prepare for the midterm too. So we're going to stop over here. This is the last topic. But even if you can prepare this diagram, I just brought this diagram to explain you the difference between the two, but this diagram is not from your book. So that's why I said here, this is like the last slide which we need to discuss. Okay, so now what is an active immunity and a passive immunity? Have you ever heard about these immunities before? What is the difference between active and the passive immunity? Anybody? No? Okay, no worry. I will explain it to you. So now it is very interesting, and please pay attention. What is an active immunity? Active immunity, when we say active, okay, that means, okay, uh, whenever my immune system is being activated, that means my B cells, okay, they they bound to an antigen, okay, and they started producing the antibodies. Okay, so that's called an active immunity. That means when your immune system is stimulated to produce the antibodies, for example. Okay, that is called as a active immunity. For example, in my case, I recently got the infection, so I have an active immunity right now because I naturally got infected with the virus, right? And then I started producing the antibodies. But then nowadays, people who did not get infected yet with the coronavirus, right? Right now, they are all are taking vaccines, right? They all are taking the Taimat, the vaccines, right? And since they're taking vaccines, it is also going to stimulate their immune system, right? But artificially, because they did not get the infection. They were stimulated. How they were stimulated? The vaccines, for example, which we get nowadays, they inject the COVID, for example, they inject the natural virus, natural bacteria, right? But a very, kind of heat killed form of it, attenuated form of it, mild form of it, right? Milder because that will simply activate their immune system, but it will not actually make them sick. Okay, so that they can continue producing the antibody. That is the main aim of vaccines, which everybody is taking right now. Okay, for example, we had in Saudi Arabia in the month of January, people who got immunizations, vaccines in the month of January, they all got Pfizer vaccines, right? You might have heard in the news as well. They all got Pfizer vaccines. So Pfizer, because nowadays how you make a vaccine is again a little different from one company to the other company. Because earlier, long back, old age methods were like, you take for example, COVID, you heat kill it, or you disarm it, for example, you break its legs, arms, everything. Okay, you make it less powerful and then inject into the patient. Okay, but then Pfizer was like, little far more, good one. Because why? Because it used simply the RNA. It took the RNA of COVID. Okay, and the people who got vaccinated with the Pfizer, they actually did not take the live virus. They simply took the RNA of the COVID. like this is like a more advanced things. And that is why you might have seen that nowadays, if people are getting vaccinated, there are some people who don't want AstraZeneca, because now in Saudi Arabia we have AstraZeneca, right? There's no Pfizer available, so the stocks are kind of finished. So some of people, they're like, no, 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 we don't want AstraZeneca, we still want to go with the Pfizer one, because you're not actually exposed to the COVID, right? And then you might have seen that, people who are getting vaccinated nowadays, they still are getting, kind of a cold, body aches and high fever and all. But it subsides, within, it subsides within 2-3 days. Okay. So it's, so that is why, kind of, they might have, like, they might get the COVID like effect for some time, for a couple of days, and after 48 hours, right, it's pretty much better. So the vaccination, the immunity which we get through the vaccination, that is basically what kind of immunity? It is the artificial immunity. Okay. So Noor says, see, it depends upon Noor. Some people, for example, my one of my brother, because it doesn't have a dead virus also, but it has, they are using AstraZeneca. I'm not sure they're using, they're using the modified version of a common cold virus, which is usually more common in chimpanzees. Okay, so they use that altered virus. Okay, so that is why if you compare AstraZeneca with the Pfizer gives you, like, 95%, you can get 95% good results from coronavirus, but AstraZeneca tells you only 84%. You should do all these surveys, right? So, but it's that, yeah, some people don't want Pfizer. For example, one of my, cousin sister, she's in Dubai. She was like, I don't want these mRNA and all those things injected in my body. I don't want this high technology things, because Pfizer used the highest technology, so I don't want this high tech technology. I want to go with, the age old technique of vaccinations. It differs from one person to the other person, right? But you might have seen that some people are nowadays still, they are waiting for Pfizer to come. They don't want to take AstraZeneca, they want Pfizer to come, because it gives you more kind of, protection even than the AstraZeneca. Got it? So that is about it. So then we have the passive immunity. Okay. In the passive immunity is like your immune system is not activated. Very important point to remember. No activation of my immune system. That means my B cells are not activated, not at all. They're not proliferated, they're not activated, right? They don't multiply. But I get something called as a ready-made, in simple thing I can explain you, like making food at home, right, from the scratch. If you make food at home, like a biryani at home, right, so that is like an active immunity, right? You made everything from the scratch. But if you order food from, or you make a phone call, I need 3 MACDs, right? So that is like, you got a ready-made food. Okay, so that is your passive immunity. You are given ready-made antibodies. Okay, so your immune system is not activated. For example, it also has two types, natural and artificial. In natural one, a baby gets a ready-made antibodies from his mother. Okay, when his mother feeds him milk, right? So he is getting ready-made antibodies, okay, by the time, and which gives him protection for the initial few months or few years. Okay, and then we have the artificial one. Artificial one is, for example, god forbid, a person a person was in a desert and he got a snake bite. Okay, and a patient, or for example, a person ate a food, with, and he got botulism because of them, because of the toxin. The botulism is, here we have a neurotoxin. Okay, so what happens, for example, if it's a snake bite, what will we do? We will simply, give, or not only snake bite, a spider bite also. Okay, so we will simply give him an injection of an anti-venom injection. What is an anti-venom injection? We will give him ready-made antibodies which we prepare right earlier, and give him the what? Give him those antibodies, so that all those antibodies which they will neutralize, okay, with the toxin, or with the snake poison, or with the neurotoxin, right? And they will neutralize it, and person can be saved as early as possible. Okay. So remember, the neurotoxin can cause the lockjaw disease. You might have studied that in biochemistry, right? The lockjaw disease, So the neurotoxin in botulism, what does it do? It prevents the release of acetylcholine from the presynaptic vesicles, right? And it can block the receptor completely. As a result, the patient can have a lockjaw disease. It will, so the muscles will be affected. You studied all that in biochemistry, right? Okay. Is it clear? And did you all understand what is the difference between the natural and the artificial immunity? So there's no chance of having a memory in the passive immunity because your immune system was not activated at all. You were given the ready-made antibodies and khalas, right? So so that is about, and those they prepare those artificial antibodies. They inject, for example, horse. They might inject a horse with the spider toxin, or with the snake toxin, right? A small amount of it. Then the horse will produce the antibodies. Okay, then they will take the blood from the horse. Okay, they will take the plasma of the horse. Okay, and then they take the serum of the horse and then isolate the antibodies and then use it. Okay, for example, if for humans, if they get the snake bite or anything like rabies, tetanus, right, hepatitis, anything, right? So those are the ready-made antibodies which we produce from the animals. Okay, is it clear? So this is like a last slide which we're going to discuss here. So are we okay with any questions till now? Okay, all clear. So as I said that on Sunday, we're not going to cancel the classes. We are going to have a class on Sunday, but the first half an hour we'll take the lymphocytes and not lymphocytes, we'll take the chapter, we'll discuss the chapter because, this is the reason why we're not canceling the classes, because we need to finish the syllabus at the same time, right? So for the second half, we can do a revision on cardiovascular system. Okay. So that's all for today. I wish you all the very best and have a wonderful and happy and studyful weekend. Yes, absolutely. I want all of you to have a studyful weekend. Study, 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 right? So, just try to get 100%, in this mid